This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to this fascinating journey through time, where we'll explore the remarkable evolution of hoofed mammals. From their humble origins millions of years ago to the diverse creatures we see today, horses, cattle, deer, and countless others, these animals have shaped ecosystems across the globe. We'll discover how simple toes transformed into powerful hooves, how ancient ancestors adapted to changing climates, and how these evolutionary innovations allowed them to thrive on every continent. Join me as we uncover the incredible story of one of nature's most successful adaptations.
Chapter 1. When Hoofed Mammals Walked with Dinosaurs. The story begins not with a spectacular skeleton mounted in a museum, but with a fur trapper's curiosity. In the early 1840s, Samuel Culbertson, working the fur trade along the Missouri River in what would become Nebraska, stumbled upon peculiar fossil bones weathering out of ancient rock layers. Unable to identify the remains, he sent them to his family in the East, who forwarded them to Joseph Leidy, a Philadelphia anatomist rapidly becoming America's foremost paleontologist. Leidy recognized the bones as belonging to extinct mammals unlike any living forms, opening a window into North America's deep past. That initial discovery hinted at a hidden history of mammalian diversity, but the truly revolutionary evidence would emerge much later and much farther away, in the Biskiti Formation of Uzbekistan. The Biskiti deposits, dating to 85 million years ago in the late Cretaceous, have yielded fragments of teeth, jawbones, and occasional limb elements that fundamentally altered our understanding of when hoofed mammals first appeared. For decades, paleontologists assumed that ungulates, the diverse assemblage including horses, cattle, deer, elephants, and whales, emerged only after the asteroid impact ended the dinosaur age 66 million years ago. The Biskiti fossils shattered that timeline, pushing ungulate origins back by 20 million years into an era when Tyrannosaurus rex still hunted and vast herds of hadrosaurs browsed Cretaceous vegetation. This discovery forced a complete reassessment of mammalian evolution during the Mesozoic, challenging the long-held assumption that early mammals remained small, nocturnal insectivores until the great reptiles fell.
The Biskiti specimens consist primarily of isolated teeth and jaw fragments rather than complete skeletons. Teeth preserve crucial information about evolutionary relationships because their complex patterns of cusps and ridges function like fingerprints, allowing paleontologists to identify species and trace lineages across millions of years. The key lies in specific molar characteristics that appear consistently across ungulate groups but remain absent in other mammalian orders. Researchers identified particular cusp arrangements and dental crests that occur in known ungulates but not in contemporary groups like primates, rodents, or carnivores. The Biskiti specimens displayed these telltale ungulate features in teeth that predated the supposed origin of the group by 20 million years. What made these teeth diagnostic involved understanding the broader context of mammalian evolution during the Cretaceous. Most mammals of this era descended from cynodonts, a group of mammal-like reptiles that had already evolved many mammalian characteristics by the late Triassic. Following the end-Triassic extinction, these early mammalian ancestors radiated into diverse forms, though they remained relatively small while dinosaurs dominated. By the late Cretaceous, mammals had diversified into numerous lineages with distinct dental patterns reflecting different diets and ecological roles. Among these Cretaceous mammals, a group called condylarths represented primitive omnivorous mammals with bunodont cheek teeth, molars featuring low, rounded cusps suitable for crushing and grinding both plant and animal matter. These animals occupied a crucial position in mammalian phylogeny, and some researchers propose them as ancestors to ungulates. The condylarths possessed relatively unspecialized skeletons that could have given rise to the diverse body plans seen in later hoofed mammals.
However, the fragmentary nature of many condylarth fossils made determining their precise relationships difficult, and the question of whether they represented true ungulate ancestors or merely close relatives remained contentious. The Bisecti discoveries complicated this picture by demonstrating that distinct ungulate lineages had already separated from other mammalian groups while condylarths still existed. This temporal overlap suggested that condylarths might represent a separate branch of mammalian evolution rather than direct ungulate ancestors, though the fragmentary fossils prevented definitive conclusions. The teeth from Uzbekistan showed features linking them unmistakably to the ungulate lineage, yet they lacked many derived characteristics that define modern ungulate groups. They represented stem lineages, evolutionary branches that diverged before living ungulate orders split from each other.
This concept of stem versus crown groups becomes crucial for interpreting what the Bisecti fossils reveal. The crown group includes the last common ancestor of all living members of a clade, plus all its descendants. For ungulates, this means the ancestor that gave rise to modern parasadactyls, odd-toed ungulates like horses, artiodactyls, even-toed ungulates like deer and cattle, and related groups, along with all species descended from that ancestor. Stened ungulates, by contrast, diverged from the main lineage before that common ancestor lived. They share many features with crown ungulates, but branched off earlier and often retain more primitive characteristics. The practical implication affects how we interpret early fossils. The Bisecti mammals demonstrate that the ungulate lineage had separated from other mammalian groups by 85 million years ago, but they don't necessarily reveal when crown ungulates originated. That event likely occurred later, possibly in the Paleocene or early Eocene. What the Bisecti fossils do establish is that the deeper evolutionary split setting ungulates on their own trajectory happened much earlier than previously suspected, during an era when dinosaurs still dominated terrestrial ecosystems. The later discovery that whales descended from even-toed ungulates would further demonstrate the remarkable evolutionary diversity within this group.
The geographic location of these fossils in Central Asia added complexity to biogeographic questions about where ungulates first evolved. Uzbekistan occupied a very different position on the globe 85 million years ago, before continents assumed their modern configuration. The ancient Tethys Ocean separated northern land masses from southern ones, creating barriers to animal dispersal. For much of the 20th century, paleontologists assumed that most major mammalian groups originated in North America or northern Eurasia, then spread to other continents. The Bisecti Formation challenged these assumptions, suggesting that Central Asian or even southern continents might have played crucial roles in early ungulate evolution that conventional narratives had overlooked. This realization forces reconsideration of mammalian ecology during the late Cretaceous. If ungulate ancestors had already diverged by 85 million years ago, what roles did they occupy in ecosystems dominated by giant herbivorous dinosaurs? The small size of most Mesozoic mammals, the Bisecti specimens suggest animals no larger than modern rats, indicates they could not compete directly with hadrosaurs and ceratopsians for the same food resources. Instead, these early ungulate relatives likely exploited niches unavailable to reptiles.
The dental evidence offers clues about ecological strategies. Some Bisecti specimens show wear patterns suggesting consumption of abrasive foods, possibly including gritty plant materials or items requiring significant processing. The development of specialized molar patterns for grinding plant matter represents a key innovation in ungulate evolution, one that would later enable descendants to dominate as large herbivores. Even in these early representatives, hints of the dental complexity characterizing later ungulates appear. The question of hooves themselves remains frustratingly unanswerable from the fragmentary Bisecti remains. The hoof represents a remarkable adaptation, converting the typical mammalian claw into a rigid, keratinous covering that protects the toe while providing a stable platform for weight-bearing. In modern ungulates, hooves facilitate rapid locomotion across firm ground. Did late Cretaceous ungulates already possess true hooves, or did this feature evolve gradually over millions of years? Hooves rarely fossilize because they consist of keratin rather than bone. Unless exceptional preservation captures soft tissues, we can only infer hoof presence from bone structure. Terminal toe bones of hoofed mammals typically show distinctive shapes reflecting the hoof covering, but isolated fragments may not preserve enough detail to identify these subtle features. Understanding these early mammals also requires considering how their ancestors survived what has been termed the nocturnal bottleneck. During the Mesozoic, mammals appear to have been primarily nocturnal, avoiding competition with diurnal dinosaurs. This nocturnal lifestyle drove the evolution of enhanced hearing and smell, both crucial for navigating darkness. The expansion of brain regions processing these senses led to overall brain size increases in mammals compared to similarly sized reptiles. The development of fur for temperature regulation, necessary because nighttime temperatures drop significantly, further distinguished early mammals from their reptilian contemporaries. These adaptations, evolved for nocturnal survival, would later prove pre-adaptive for other lifestyles once dinosaurs disappeared. The acute hearing and sophisticated brain processing that helped Cretaceous mammals hunt insects at night could be redirected toward detecting predators or communicating with conspecifics in diurnal environments. The thermoregulatory abilities conferred by fur and endothermic metabolism allowed mammals to remain active in conditions that would incapacitate cold-blooded reptiles. The bisecti ungulates, though living alongside dinosaurs, carried within their bodies the evolutionary toolkit that would enable their descendants to radiate explosively after the asteroid impact. Determining evolutionary relationships among these early ungulates remains contentious despite molecular analyses that have clarified many aspects of mammalian phylogeny. While genetic evidence strongly supports the placement of major ungulate groups within the larger mammalian tree, fossils alone often cannot resolve relationships with certainty. The tension between molecular and morphological evidence creates ongoing debates about classifications. Molecular data derives from living animals, comparing DNA sequences to construct phylogenetic trees based on mutation rates and genetic similarities. This approach has revolutionized our understanding of mammalian relationships, revealing unexpected connections like the close relationship between whales and hippos, or the grouping of elephants with manatees. However, fossils can only be classified using morphological features, skeletal anatomy, tooth structure, and other physical characteristics. Sometimes these morphological interpretations conflict with molecular phylogenies. When molecular evidence places two groups as close relatives, but their skeletal anatomy appears quite different, paleontologists face the challenge of explaining why morphology misled them. Conversely, striking anatomical similarities sometimes result from convergent evolution rather than shared ancestry, misleading researchers who lack molecular data to correct their interpretations. The bisecti specimens, fragmentary as they are, cannot definitively answer questions about which modern ungulate groups shared more recent common ancestry. The fossils anchor one end of a timeline extending to the present, but the vast gap between 85-million-year-old fragments and well-documented Eocene ungulates spans some 20 million years, coinciding with dramatic events including the asteroid impact that triggered mass extinction. Connecting these evolutionary dots across this chasm while accounting for the geographic spread of early ungulates across multiple continents requires careful integration of fossil, molecular, and biogeographic evidence. As paleontologists continue excavating late Cretaceous deposits in Central Asia and elsewhere, each new discovery has potential to further transform our understanding. The field stands at a fascinating juncture, where traditional morphological analysis intersects with molecular phylogenetics, biomechanical modeling, and isotopic analysis of ancient tissues. Each approach provides different insights, and reconciling their sometimes contradictory conclusions requires careful evaluation of evidence from multiple sources. The bisecti fossils established that ungulate lineages had diverged while dinosaurs still dominated, fundamentally revising the narrative of mammalian evolution from one of sudden post-extinction radiation to a more nuanced story of Mesozoic diversification, setting the stage for Cenozoic dominance. Chapter 2: Climate Change Driving Post-Extinction Mammal Diversification and Ecosystem Recovery Patterns. The asteroid struck with the force of 1 billion atomic bombs. Within hours, debris from the impact site in what is now Mexico's Yucatan Peninsula had circled the globe, igniting fires across entire continents. The blast wave flattened forests thousands of miles from ground zero. But the immediate destruction, catastrophic as it was, paled in comparison to what followed. Dust and soot choked the atmosphere, blocking sunlight for months or possibly years. Photosynthesis ground to a halt across vast swaths of the planet. Global temperatures, which had averaged perhaps 25 degrees Celsius in many regions, plummeted by an estimated 30 degrees within weeks. The Earth plunged into an impact winter that would last years, perhaps decades. When the skies finally cleared 66 million years ago, the world bore little resemblance to what had existed before. The great herbivorous dinosaurs that had dominated terrestrial plant consumption for over 100 million years were gone. The massive carnivores that had preyed upon them vanished as well. Ecosystems collapsed in cascading waves as species dependent on these keystone animals went extinct. Yet amid the devastation, survivors clung to existence. Small mammals that had spent the Cretaceous period as marginal players in ecosystems controlled by reptiles found themselves in a transformed world with vacant ecological roles and no large competitors. The story of how these survivors rebuilt the biosphere emerges from an unexpected source, a series of fossil beds in the Denver Basin of Colorado that preserve an unusually complete record of the first million years after the mass extinction. Unlike most fossil deposits, which capture snapshots separated by millions of years, these Colorado formations document recovery in remarkably fine temporal resolution. Paleontologists working at sites like Coral Bluffs discovered layer upon layer of rock containing not just mammal bones but also plant fossils and volcanic ash deposits that could be precisely dated using radiometric techniques. Tyler Lyson and his team spent years excavating these beds, carefully noting the exact stratigraphic position of each fossil. The volcanic ash layers proved crucial because they contained minerals whose radioactive decay could be measured with extraordinary precision, providing ages accurate to within tens of thousands of years. This level of resolution, unheard of for deposits of this age, allowed researchers to track changes in mammalian diversity and body size across the critical first million years of recovery with unprecedented detail. The picture that emerged challenged simplistic narratives of steady, gradual recovery. Instead, the fossil record revealed a punctuated pattern of rapid diversification and body size increase, with each pulse coinciding with environmental changes. In the immediate aftermath of the extinction, surviving mammals were tiny creatures, most weighing less than half a kilogram. These rat-sized animals represented the few lineages that had managed to persist through the impact winter, likely by consuming seeds, insects, or detritus that didn't require active photosynthesis. Within the first 100,000 years after the extinction, something remarkable occurred. Mammalian species richness doubled. Where only a handful of species had scratched out existence in the barren post-impact landscape, now dozens of distinct forms appeared in the fossil record. Body sizes increased dramatically as well, with some species reaching 7 kilograms, 14 times heavier than the immediate survivors. This represents an explosive radiation by geological standards, all compressed into a time span equivalent to less than 2% of the interval between the asteroid impact and the present day. What drove this initial burst of diversification? The fossil evidence points to the recovery of plant communities. The ash and debris that had smothered photosynthesis settled out within years, but the ecosystem damage persisted much longer. The first plants to recolonize devastated landscapes were pioneer species, ferns, horsetails, and other rapid colonizers that could exploit disturbed ground. These early successional communities provided little food for herbivores, consisting largely of species with toxic compounds or poor nutritional value. The Colorado plant fossils document a gradual transition from these impoverished pioneer communities to more complex forests. As soil recovered and climate stabilized, flowering plants began to establish themselves. These angiosperms, the group that includes most modern trees, shrubs, and herbs, had been diversifying throughout the Cretaceous, but really came into their own in the Paleocene. Their leaves, fruits, and seeds provided far more diverse food sources than the fern prairies that initially dominated. This re-establishment of forest ecosystems created new niches for mammals. Species could specialize on different plant parts, some consuming leaves, others fruits, still others seeds or bark. The vertical structure of forests added three-dimensional complexity, with opportunities for animals adapted to life in the canopy, the understory, or the forest floor. As plant diversity increased, so did the number of mammalian species that could coexist by partitioning resources. Yet the tempo of this recovery varied dramatically over time, and here the Colorado fossils revealed something unexpected. The researchers identified three distinct warming intervals during the first million years after the extinction, each lasting tens of thousands of years. These weren't minor temperature fluctuations, but significant shifts that fundamentally altered the environment. During each warming pulse, both plant diversity and mammalian diversity increased markedly. The first warming occurred roughly 150,000 years after the impact. Temperatures, which had cooled considerably after the initial impact winter, rose by approximately 2 degrees Celsius. This might seem modest, but at the cooler baseline of the early Paleocene, such a shift represented the difference between marginal conditions for forest development and robust tree growth. The fossil plant assemblages from this interval show increased diversity and abundance of warm-adapted species. Mammal fossils from the same layers document the appearance of new species and the first significant size increases. A second warming interval came around 300,000 years post-impact. This pulse proved even more consequential for mammalian evolution. During this period, the largest mammals reached 15 kilograms, 30 times heavier than the immediate post-extinction survivors, and more than double the maximum size from the first 100,000 years. These animals approached the size of modern badgers or small dogs, a far cry from the rat-sized creatures that had persisted through the impact winter. This dramatic size increase reflects a fundamental principle of mammalian energetics. Larger body size requires more total energy, but it also confers advantages in terms of defense against predators, ability to process lower quality food, and capacity to survive lean periods. In the recovering Paleocene ecosystems, the expanding forests provided sufficient food resources to support these larger herbivores for the first time since the extinction. The bigger mammals could exploit coarser plant materials that smaller species couldn't efficiently process, opening new dietary niches. The third and most dramatic warming occurred around 700,000 years after the extinction, and it coincided with a pivotal event in plant evolution, the appearance and spread of the legume family. Legumes, which today include beans, peas, clover, and many trees, possess a remarkable adaptation. Their roots form symbiotic relationships with bacteria that can fix atmospheric nitrogen, converting it into forms plants can use. This allows legumes to thrive in nitrogen-poor soils where other plants struggle, and their nitrogen-rich tissues provide exceptional nutrition for herbivores. The bigger mammals could exploit coarser plant materials that smaller species couldn't efficiently process, opening new dietary niches. The fossil record from coral bluffs shows legumes appearing suddenly around this time, and their impact on mammalian evolution proved immediate and profound. Within a relatively short interval, mammals reaching 50 kilograms appeared, the size of a large dog or small wolf. These animals represented a completely different magnitude of herbivore than anything that had existed in the first 500,000 years of recovery. Their teeth show adaptations for processing tough plant materials, and their leg bones suggest they were capable runners, the first hints of the cursorial adaptations that would later characterize many ungulate lineages. The correlation between warming events and evolutionary pulses raised an obvious question: what caused these temperature increases? The answer appears to lie in volcanic activity on the Indian subcontinent. The Deccan Traps, an immense sequence of lava flows covering much of what is now west-central India, had been erupting for hundreds of thousands of years when the asteroid struck. These weren't typical volcanic eruptions, but massive flood basalt events that poured molten rock across thousands of square kilometers, releasing enormous quantities of carbon dioxide into the atmosphere. For decades, some researchers argued these eruptions, rather than the asteroid impact, caused the mass extinction. Computer simulations ultimately settled this debate in favor of the asteroid hypothesis. The volcanic emissions did warm the climate, but only by about 2 degrees Celsius, significant but insufficient to trigger the catastrophic ecosystem collapse that occurred at the Cretaceous-Paleogene boundary. The asteroid's impact winter, dropping temperatures by 30 degrees, could and did cause mass extinction. The volcanic warming, by contrast, was too gradual and too small in magnitude. However, those same volcanic warming pulses that proved insufficient to cause extinction turned out to be perfectly calibrated to drive post-extinction recovery. After the world had cooled from the impact winter and begun to stabilize, the periodic warming events from Deccan volcanism provided exactly the climate boost needed to accelerate forest recovery. Warmer temperatures extended growing seasons, increased precipitation in many regions, and allowed warmth-loving plants to expand their ranges. This creates a fascinating irony. The Deccan Traps eruptions, once proposed as the cause of the mass extinction, actually facilitated recovery from that extinction. The very process that some scientists had blamed for ending the Cretaceous world instead helped establish the Paleocene ecosystems where modern mammalian groups first diversified. Timing proved critical. Had the volcanic warming occurred during the impact winter, it might have slightly ameliorated the temperature crash. But volcanic warming taking place after the immediate crisis had passed served as a catalyst for ecosystem rebuilding. Warmer temperatures extended growing seasons, increased precipitation in many regions, and allowed warmth-loving plants to expand their ranges. This creates a fascinating irony. The Deccan Traps eruptions, once proposed as the cause of the mass extinction, actually facilitated recovery from that extinction. The very process that some scientists had blamed for ending the Cretaceous world instead helped establish the Paleocene ecosystems where modern mammalian groups first diversified. Timing proved critical. Had the volcanic warming occurred during the impact winter, it might have slightly ameliorated the temperature crash. But volcanic warming taking place after the immediate crisis had passed served as a catalyst for ecosystem rebuilding. The forests that developed during these warming intervals bore little resemblance to modern temperate woodlands. Paleobotanical evidence from the Denver Basin and elsewhere indicates these were dense, multi-layered assemblages dominated by flowering plants rather than the conifer-dominated forests of the Cretaceous. Palm fossils appear far north of their modern range, suggesting subtropical conditions extended into what is now the northern United States. The absence of clear growth rings in many fossil trees indicates year-round growing conditions without harsh seasonal dormancy. These perpetually productive forests could support higher animal densities than the seasonal environments that came later. A mammal population limited by winter food scarcity can maintain far fewer individuals than one with year-round access to resources. The lack of harsh seasonality in the early Paleocene meant mammalian populations could grow larger, creating more opportunities for speciation as populations fragmented across the landscape. The relationship between plants and mammals extended beyond simple provision of food.
Mammals served as seed dispersers for many plant species, carrying fruits away from parent trees and depositing seeds in new locations, along with a convenient packet of fertilizer. This mutualism would become increasingly important as larger mammals capable of dispersing larger seeds evolved. Some paleobotanists argue that the evolution of large mammalian herbivores and the diversification of large fruited plants represent a co-evolutionary process, each group shaping the evolution of the other.
By 1 million years after the extinction, the Coral Bluffs fossil beds document ecosystems that would have seemed almost unrecognizable compared to the devastated post-impact landscape. Instead of a few species of small, generalized mammals, dozens of distinct forms occupied varied ecological roles. Some had teeth adapted for eating leaves, others for processing fruits or seeds, still others for consuming insects or other animals. Body sizes ranged from mouse-sized insectivores to badger-sized herbivores capable of defending themselves against most predators. Yet these recovering ecosystems still differed fundamentally from what would come later. The largest mammals remained modest in size compared to the dinosaurs they replaced or the giant mammals that would evolve later in the Cenozoic. No mammalian predators approached the size of the great tyrannosaurs, and no herbivores matched the bulk of sauropods or hadrosaurs. The Paleocene fauna consisted of mid-sized animals exploiting niches that had been peripheral during the age of dinosaurs. The climate-driven pattern of diversification documented in Colorado likely played out across the globe, though few other locations preserve such detailed records. Fossil beds in Mongolia, France, and Montana show similar trends of increasing mammalian diversity and body size through the Paleocene, though without the temporal resolution to identify specific warming pulses. The global nature of these changes suggests environmental forcing operating at a planetary scale, consistent with worldwide volcanic warming driving ecosystem recovery. Understanding this climate-mediated recovery pattern transforms our interpretation of early mammalian evolution. Rather than a simple competitive release where mammals automatically filled vacant niches after dinosaur extinction, what actually occurred was a complex interaction between climate change, plant community development, and mammalian adaptation. Each warming pulse opened new possibilities. Each forest recovery allowed new species to evolve. Each increase in mammalian diversity fed back to influence plant evolution through pollination and seed dispersal. The Colorado fossil beds also demonstrate the importance of environmental context for interpreting evolutionary change. Without the climate data from volcanic dating and paleobotanical reconstructions, we might look at the pattern of mammalian diversification and attribute it to internal evolutionary processes, competition driving specialization perhaps, or intrinsic limitations on how quickly new species can evolve. Instead, the evidence clearly shows external environmental changes pacing evolutionary innovation. This raises profound questions about what drives evolutionary patterns more broadly. How much of the vast diversity of modern mammals reflects responses to changing climate and vegetation over the past 66 million years? What role did subsequent climate fluctuations play in generating the incredible array of ungulate forms that would eventually evolve, from tiny mouse deer to massive elephants, from fleet horses to aquatic hippos? Did each major climate shift trigger a new pulse of adaptation and diversification? Or did mammals eventually achieve sufficient ecological dominance to buffer themselves against environmental change? Chapter 3. Whales evolved from hoofed land mammals through dramatic aquatic adaptations. Picture a wolf prowling the banks of shallow rivers in what is now Pakistan, 53 million years ago. The creature stands about the size of a modern gray wolf, with 4 legs ending in hooves rather than paws. Its elongated skull houses eyes positioned higher on the head than typical terrestrial predators, closer to the top like those of a crocodile. This animal, called Pakasitis, spent much of its time wading through water hunting for fish, and it represents one of the most extraordinary evolutionary transformations documented in the fossil record, the ancestor of whales. The proposal that whales descended from hoofed land mammals initially struck many scientists as absurd when first suggested in the 19th century. How could a terrestrial quadruped with legs designed for walking on solid ground give rise to the largest animals ever to exist, creatures so supremely adapted to aquatic life that they would die if stranded on a beach? The transformation seemed to violate basic principles of adaptation, requiring wholesale reorganization of nearly every body system. Yet, the accumulation of fossil evidence over the past several decades has demonstrated beyond reasonable doubt that this improbable transition occurred, and in surprisingly rapid geological time. The story begins not with whales, but with their unexpected cousins. Molecular analyses comparing DNA sequences across mammalian groups revealed something shocking. The closest living relatives of whales are not seals, sea lions, or any other aquatic mammals, but hippopotamuses. This finding, published in the late 20th century, met with considerable skepticism from paleontologists who could see no obvious anatomical link between hippos and cetaceans. Hippos, after all, possess 4 legs, live partially on land, and show none of the extreme aquatic specializations characteristic of whales. How could genetic evidence suggest such a close relationship when morphology seemed to argue against it? The resolution came from fossils discovered in Eocene deposits of Pakistan during the 1970s and 1980s. These revealed a series of animals intermediate between fully terrestrial mammals and obligately aquatic whales, documenting a transformation that played out over roughly 15 million years. The fossils showed that whales belonged to the artiodactyls, the even-toed ungulates that include pigs, deer, cattle, and camels. More specifically, cetaceans and hippos together form a branch of the artiodactyl tree, meaning that whales are not just related to hoofed mammals, but are themselves modified hoofed mammals. This classification required acceptance of a peculiar fact. A blue whale, despite lacking anything resembling hooves and living its entire life in the ocean, shares more recent common ancestry with a cow than a cow shares with a horse. The ankle bone, called the astragalus, provided crucial evidence. In artiodactyls, this bone has a distinctive double pulley shape, unlike that found in any other mammalian group. Early whale fossils from Pakistan possessed this characteristic artiodactyl astragalus, demonstrating their membership in the even-toed ungulate clade despite their aquatic adaptations. But which artiodactyls gave rise to whales? For over a century, scientists debated this question, with many favoring the mesonychids as ancestral candidates. These extinct carnivorous ungulates possessed triangular teeth remarkably similar to those of early whales, suggesting a close relationship. The teeth of both groups featured serrated edges suited for slicing meat, and their overall dental pattern seemed too similar to represent convergent evolution. Mesonychids themselves were thought to have descended from arctocyaenids, raccoon-like omnivores that appeared shortly after the dinosaur extinction. This hypothesis collapsed under molecular evidence, demonstrating that whales nested within artiodactyls rather than representing a sister group to them. If whales arose from within the even-toed ungulates, they could not have descended from mesonychids, which branched off earlier. The triangular teeth, so long considered proof of mesonychid ancestry, instead represented convergent evolution, separate lineages independently evolving similar dental patterns for processing meat. The true ancestors emerged from an unexpected source. Researchers working in northern India and Pakistan discovered fossils of the Rowellid family, small herbivorous mammals that lived during the Eocene. These sheep-sized animals showed a constellation of features linking them to whale origins. Most dramatically, they possessed a thickened bone in the middle ear called the involucrum, a structure found only in cetaceans and nowhere else in the mammalian world. This involucrum functions in modern whales to enhance underwater hearing, suggesting that Rowellids had already begun adapting to aquatic environments. Indohyus, the most thoroughly studied Rowellid, reveals an animal caught between terrestrial and aquatic lifestyles. Its limb bones show unusual thickness and density, characteristics shared by modern aquatic mammals like manatees and hippos. This bone density reduces buoyancy, allowing animals to walk along river bottoms rather than floating helplessly at the surface. Behavioral observations of modern water chevrotains, small deer-like animals, provide a probable analog for Indo-Hyas behavior. When threatened by predators, water chevrotains flee into streams and hide underwater for up to 4 minutes, browsing on aquatic vegetation until danger passes. Indo-Hyas likely employed similar tactics, using water as refuge rather than primary habitat.
The isotope chemistry of Indo-Hyas teeth confirms this semi-aquatic lifestyle. By analyzing the ratio of different oxygen isotopes preserved in tooth enamel, researchers can determine what kind of water an animal drank during life. Indo-Hyas consumed fresh water exclusively, indicating it lived near rivers and streams rather than in marine environments. Its diet appears to have consisted of land plants, though some specimens show evidence of consuming aquatic vegetation as well. From this semi-aquatic starting point, the transition to fully aquatic existence proceeded through several distinct stages, each documented by fossil forms from Pakistan's Eocene deposits. Pakicetus represents the first stage, an animal still primarily terrestrial but showing initial adaptations for aquatic hunting. Its eyes, positioned high on the skull, allowed it to float at the water surface with most of its body submerged, watching for prey above the waterline like a crocodile. The ears, however, remain terrestrial in design, unable to hear directionally underwater. Pakicetus probably hunted by ambush, lurking in shallow water and lunging at animals that came to drink. The skeleton of Pakicetus reveals an animal built for running and jumping on land, not swimming. Its leg proportions match those of terrestrial predators, with long lower leg bones providing leverage for rapid acceleration. The feet bore small hooves on each toe, confirming its membership in the ungulate clade. Yet certain skull features already diverged from typical land mammals. The elongated snout and closely spaced forward-facing eyes gave it a superficially crocodile-like appearance, though the underlying bone structure remained fundamentally mammalian. Approximately 4 million years later, around 49 million years ago, animals like Ambulocetus appeared. The name translates as walking whale, and these creatures truly straddled the boundary between land and water. Ambulocetus stretched about 3 meters long, with a powerful tail and reduced but still functional legs. The forelimbs were shorter relative to body size than in Pakicetus, while the hind limbs became elongated and likely webbed, judging from the bone structure. How did Ambulocetus move? Biomechanical analysis suggests it swam using pelvic paddling, much like modern otters. The powerful hind limbs provided propulsion through alternating kicks, while the tail contributed additional thrust through undulation. On land, movement would have been laborious and awkward, similar to modern seals struggling across beaches. The vertebral column shows adaptations restricting lateral flexibility while enhancing dorsoventral bending, the opposite of most land mammals but ideal for aquatic undulation. The hearing apparatus of Ambulocetus reveals partial adaptation to underwater sound detection. While lacking the sophisticated acoustic isolation of modern whales, the skull shows initial modifications allowing some directional hearing in water. The jaw began developing a fat pad in the mandibular foramen, a channel in the lower jaw that in modern toothed whales houses fatty tissue conducting sound vibrations to the ear. This represented a crucial step toward the advanced echolocation systems that would evolve later. Remingtonocetids, living around 48 million years ago, pushed aquatic adaptation further. These animals possessed even more reduced limbs and elongated bodies, suggesting they rarely, if ever, came onto land. Their skulls show eyes positioned laterally rather than on top, indicating they hunted fully submerged rather than floating at the surface. The nasal opening remained near the tip of the snout, not yet migrated backward toward the top of the head as in later whales. Isotope analysis demonstrates that some Remingtonocetid species consumed only marine water, having lost the dependency on fresh water that characterized earlier forms.
This represents a fundamental ecological transition, from animals tied to rivers and coastal regions to creatures capable of exploiting the open ocean. The semicircular canals of the inner ear, crucial for balance in terrestrial mammals, had already begun to shrink in Remingtonocetids. This reduction accompanied their increasingly aquatic lifestyle, as the three-dimensional vestibular sense, so important on land, became less critical in water where buoyancy provides support. The protocetids, appearing around 47 million years ago, included both coastal and fully marine species. Some, like Myocetus, still gave birth on land, as evidenced by a fossil of a pregnant female whose fetus was positioned head first for terrestrial delivery. Had Myocetus given birth in water, the fetus would have been positioned tail first to prevent drowning during the birth process. Other protocetids had achieved sufficient aquatic competence to venture far from shore, with fossils appearing in deep marine deposits. Rhodhocetus, one of the best-known protocetids, illustrates the profound skeletal changes accompanying full aquatic commitment. The sacrum, a fusion of vertebrae connecting the pelvis to the spine in land mammals, had begun to separate into individual vertebrae, indicating loss of the rigid pelvic spinal connection necessary for terrestrial locomotion. The hind limbs remained present but reduced, no longer bearing the animal's weight. The tail vertebrae elongated and likely supported a horizontal fluke, though direct evidence of flukes rarely fossilizes. By 35 million years ago, basilosaurids represented fully obligate marine whales, incapable of supporting their weight on land. These serpentine creatures stretched up to 18 meters long, with tiny vestigial hind limbs completely disconnected from the spine. The forelimbs had transformed into rigid flippers, retaining the finger bones of their land mammal ancestors but encasing them in an inflexible paddle. The elongated vertebral column undulated dorsoventrally rather than laterally, generating thrust through vertical tail motions rather than the lateral body waves of fish. The nasal opening had migrated to the top of the skull, forming a primitive blowhole. This allowed breathing while the body remained submerged, an essential adaptation for an air-breathing animal spending its entire life in water. The skull shows elaborate sinuses isolating the ear from surrounding tissues, enabling the directional hearing underwater that would later develop into echolocation in toothed whales. The teeth, however, remained heterodont, differentiated into incisors, canines, and molars like land mammals, rather than the uniform conical teeth of modern toothed whales. Basilosaurids lacked a melon organ, a mass of fatty tissue in the forehead of modern toothed whales that focuses sound for echolocation. Their brains, smaller relative to body size than in modern cetaceans, suggest limited cognitive abilities compared to later forms. They probably hunted by vision and perhaps limited acoustic detection, pursuing fish and squid in coastal and open ocean waters. Stomach contents of one specimen revealed fish bones, confirming a diet of marine prey. The Durodontines, close relatives of basilosaurids living during the same period, show different body proportions, more similar to modern whales. Rather than the extreme elongation characterizing basilosaurids, Durodontines maintained more compact bodies with powerful tail flukes. They swam using caudal oscillation, the up-down tail motion employed by all modern whales, rather than the whole-body undulation of basilosaurids. This more efficient swimming style allowed greater speed and maneuverability, probably contributing to the success of their descendants. From these Eocene forms evolved the two modern cetacean lineages, the toothed whales or odontocets, including dolphins, porpoises, and sperm whales, and the baleen whales or mystacets, the filter-feeding giants like blue whales and humpbacks. The split between these groups occurred around 34 million years ago, coinciding with global climate cooling and the formation of Antarctic ice sheets. This environmental change altered ocean circulation patterns and nutrient distribution, creating opportunities for filter-feeding specialists to exploit dense concentrations of small prey. The evolution of baleen, the comb-like plates of keratin that mysticetes use to strain food from seawater, represents one of the most remarkable adaptations in mammalian history. Early masticites possessed both teeth and rudimentary baleen, suggesting a gradual transition. As baleen became more elaborate, teeth reduced and eventually disappeared entirely. Multiple genes responsible for tooth enamel formation accumulated mutations in baleen whale lineages, converting functional genes into non-functional pseudogenes. This genetic evidence confirms that tooth loss occurred after the masticity odontoset split, with toothed ancestors giving rise to toothless descendants. The size increase in baleen whales directly correlates with baleen development and filter feeding efficiency. Larger whales can support larger baleen plates, allowing them to filter more water and capture more prey per mouthful. This created a positive feedback loop. Mutations increasing body size improved feeding efficiency, and improved feeding efficiency allowed support of larger body sizes. The blue whale, at up to 30 meters long and 200 tons in weight, represents the culmination of this trend, the largest animal ever to exist on Earth, marine or terrestrial, living or extinct. Toothed whales evolved along a different trajectory, developing echolocation that allowed them to hunt in darkness and pursue fast-moving prey. The melon organ focuses outgoing clicks into a directional beam of sound, while specialized jawbones and associated fatty tissues receive returning echoes. This biological sonar surpasses human-engineered systems in many respects, allowing dolphins to detect fish buried in sediment or distinguish between objects differing by millimeters in size. The biogeographic origin of these transformations remains somewhat uncertain. Most of the crucial transitional fossils come from Pakistan and northern India, suggesting South Asia as the primary theater of whale evolution. However, the Indian subcontinent occupied a very different position 50 million years ago, still separated from the Asian mainland by the Tethys Ocean. Some researchers propose that cetaceans evolved in isolation on the Indian landmass, only dispersing to other regions after India collided with Asia around 45 million years ago. Alternative scenarios place the origin in Africa or the Middle East, regions connected to the Tethys Ocean during the Eocene. Fragmentary fossils from Egypt and the Arabian Peninsula hint at early cetacean presence in these areas, though the specimens remain less complete than those from Pakistan. The global distribution of protocetids and basilosaurids by the Middle Eocene demonstrates that whales achieved worldwide dispersal relatively quickly, once committed to marine existence. What environmental pressures drove this extraordinary transition? Competition with terrestrial predators may have pushed ancestral artiodactyls toward aquatic refuges, where their semi-aquatic habits provided advantages. Alternatively, abundant aquatic food resources, fish, aquatic invertebrates, and marine mammals, might have lured increasingly specialized feeders into permanent marine residence. The enormous productivity of Eocene oceans, warmed by greenhouse conditions and enriched by nutrient cycling from rapid plate tectonic activity, would have supported large populations of marine prey. The rapidity of the land-to-sea transition suggests strong selective pressure favoring aquatic specialization. From Pachycetus to Basilosaurids represents less than 20 million years, a geological instant compared to the hundreds of millions of years over which whales' fish competitors evolved their streamlined forms. This compressed time frame implies that intermediate stages faced powerful selection to either complete the transition or abandon it, with partial adaptation conferring sufficient advantages to drive rapid change. Yet mysteries remain. How did air-breathing mammals solve the diving challenge, allowing them to pursue prey to depths where water pressure would collapse land mammal lungs? Modern deep-diving whales collapse their lungs intentionally, storing oxygen in blood and muscle rather than air spaces. The physiological changes enabling this capability must have evolved gradually, but the fossil record cannot reveal soft tissue adaptations like myoglobin concentrations or cardiovascular modifications. Somewhere between Ambulocetus, probably limited to shallow dives, and modern sperm whales capable of descending 3 kilometers deep, this transformation occurred. When and through what intermediate stages remains unknown. Chapter 4.
Genes reveal hidden unity. Stand a male white-tailed deer next to a domestic bull, and the differences seem absolute. The deer's antlers branch into multiple times, each point covered in fuzzy velvet during growth that later peels away to reveal bare bone. Each autumn, these magnificent structures drop off entirely, only to regrow the following spring in an annual cycle of shedding and regeneration. The bull's horns, by contrast, emerge as permanent fixtures, wrapped in a keratin sheath that continues growing throughout the animal's life, never branching, never shedding. Place a giraffe beside these two, and the distinctions multiply further. Its ossicones remain covered in skin and hair, looking less like weapons and more like permanent bumps on the skull. For over a century, biologists looking at these stark differences concluded that each type of cranial appendage must have evolved independently. How could structures so fundamentally distinct in composition, growth pattern, and life cycle share a common origin? This interpretation, termed the essential integument concept, treated the outer covering, whether keratin sheath, velvet and bare bone, or skin and hair, as an integral component of each structure's identity. Under this framework, a horn without its keratin sheath would not be a horn at all, just as an antler that never shed its velvet would fail to meet the definition of a true antler.
Yet this interpretation faced a troubling problem. Every ruminant cranial appendage, regardless of its outer covering, shares a fundamental commonality. All grow from paired bony outgrowths positioned near the frontal and parietal bones of the skull. The homologous core hypothesis proposed instead that all descended from a single rudimentary bony outgrowth in the common ancestor of all ruminants, with the various coverings representing later elaborations that evolved independently in different lineages. The hypothesis treated the permanent bone portion as the ancestral structure and the integument as secondary, much like considering fur color patterns as modifications to a shared mammalian hair covering rather than as defining characteristics. For decades, the debate remained anchored in morphology and fossils. Then, advances in RNA sequencing technology made it possible to identify which genes are active in developing tissues, providing a window into the molecular programs that build complex body structures. If horns and antlers truly shared a common developmental origin, their growing tissues should activate similar sets of genes, despite their obvious physical differences. Zachary Calamari at the American Museum of Natural History designed an experiment to test this prediction. His team focused on juvenile cattle developing their first horns and compared gene expression patterns in these horn buds to those in developing deer antlers. As a phylogenetic outgroup for comparison, a reference point representing tissues that definitely did not share cranial appendage origins, they used skin samples from pigs, close relatives of ruminants that lack any form of cranial appendage. The challenge lay in obtaining appropriate tissue samples. Horn buds in cattle begin differentiating from surrounding tissues during the second month of fetal development, but actual growth of the bony core does not occur until after birth. The team needed horn bud tissue actively engaged in bone formation, not merely tissue that had received its developmental fate but not yet begun growing. Similarly, the antler samples required tissue from the growth phase, when cells were rapidly dividing and ossifying. The researchers extracted RNA from these tissues and sequenced it to determine which genes were being transcribed, effectively reading out the molecular recipe each tissue was following to build its structure. The resulting transcriptomes provided snapshots of cellular activity, revealing not just which genes were turned on, but how strongly they were being expressed. Sophisticated computational analyses then compared these gene expression patterns across the different tissue types, identifying genes that showed similar activity in horn buds and antlers while differing significantly in pigskin. The results challenged the essential integument concept decisively. Despite the profound differences in how horns and antlers grow and function, their developing tissues showed remarkably similar gene expression patterns. Numerous genes involved in bone formation and cellular development were activated at comparable levels in both horn buds and developing antlers, while remaining inactive or expressed at very different levels in pigskin. These differentially expressed genes provided molecular evidence for a shared developmental program, suggesting that horns and antlers indeed descended from a common ancestral structure. More striking still, the analysis revealed which cells gave rise to cranial appendages. The gene expression patterns pointed unmistakably to the cranial neural crest, a transient population of cells unique to vertebrate embryos. During early development, the neural crest forms along the edges of the neural tube, the structure that will become the brain and spinal cord. These remarkable cells then migrate throughout the embryo, settling in various locations and giving rise to an astonishing diversity of tissues. In the head, cranial neural crest cells construct much of the facial skeleton. They form the bones of the upper and lower jaw, the bones surrounding the eyes, and various other elements of the skull. These cells possess unusual developmental plasticity, capable of generating both bone and cartilage depending on the signals they receive from surrounding tissues. The discovery that horn and antler tissues bore the molecular signature of cranial neural crest origin placed them firmly within this developmental program. This finding carried profound implications. The bones on the sides and back of the mammalian skull, the parietals, temporals, and occipitals, derive not from neural crest but from mesoderm, a different embryonic tissue layer. If cranial appendages grew from these mesodermal bones of the skull vault, their gene expression patterns should resemble those of other mesodermal skeletal elements. Instead, the neural crest signature linked cranial appendages to the facial skeleton rather than to the brain case, suggesting they evolved through modification of facial development programs rather than through simple outgrowth from the skull roof. This distinction matters because it reveals that cranial appendages represent elaborations of the same cellular machinery that builds jaws and other facial structures, not extensions of the bones protecting the brain. The molecular similarities between horns and antlers existed despite striking differences in the tissues that appear to initiate their growth. Horn development seemingly begins in specialized dermis and subcutaneous tissue overlying the frontal bone. This region, sometimes called the os cornu, shows dramatic increases in nerve fibers and other structural changes in developing horn buds compared to ordinary skin covering the rest of the skull. The specialized tissues promote bone growth through intramembranous ossification, the process by which bone forms directly from connective tissue without a cartilage intermediate stage. Antler growth follows a different pattern. The periosteum, a membrane covering the outer surface of bones, initiates pedicle development. The pedicle is the permanent bony base from which antlers grow and regenerate. Early pedicle formation occurs through intramembranous ossification like horn growth, but later stages of pedicle development and all subsequent antler growth from the pedicle proceed through endochondral ossification. This process first forms a cartilage model that is then gradually replaced by bone, the same mechanism that builds most of the mammalian skeleton. These developmental differences once seemed to argue conclusively against horn-antler homology. How could structures that grow from different tissue layers and employ different ossification processes share a common origin? The gene expression data suggested an answer. Perhaps these tissue differences represent later modifications to a shared developmental program rather than evidence of completely separate origins. The initial stages of both structures employ intramembranous ossification and occur in similar locations on the skull. The later switch to endochondral ossification in antlers and the apparent shift in which tissue layer drives growth might have evolved after the antler lineage split from the horn lineage. Recent research on horn development added an intriguing complication. Evidence suggested that the horn bud, the specialized tissue that promotes bone growth, and the keratin sheath that ultimately covers the horn might receive their developmental fates separately during embryonic development. If true, this would mean the bony core and its keratin covering are not intrinsically linked developmental units, but rather separately determined structures that happen to grow in association with each other. The keratin sheath might represent a secondary addition to an ancestral bony outgrowth, exactly as the homologous core hypothesis proposed. This finding would explain how the essential integument could vary so dramatically across ruminant families: horn sheaths, velvet coverings, and skin-covered ossicones, while the underlying bony cores employed the same cranial neural crest-derived developmental program. The phylogenetic relationships among ruminant families introduced further complexity to the homology question. Molecular analyses consistently placed Moschidae, the musk deer, which completely lack cranial appendages, as the closest relatives of Bovidae, the horn-bearing family that includes cattle, antelopes, and goats. This nested position of a cranial appendage-lacking family within the radiation of cranial appendage-bearing families created a taxonomic puzzle. If cranial appendages evolved once in the common ancestor of all ruminants, then Moschidae must have secondarily lost this feature, their ancestors having once possessed some form of cranial appendage that was later abandoned. Alternatively, if cranial appendages evolved multiple times, the placement of Moschidae simply represented one lineage that never acquired the trait. However, the gene expression evidence supporting a shared origin for horns and antlers made multiple independent origins increasingly difficult to defend. The more parsimonious explanation held that the ancestral ruminant possessed a rudimentary bony cranial outgrowth, that Moschidae lost this structure, and that the various modern forms represent elaborations on this ancestral theme. The fossil record of Moschidae remains sparse regarding cranial appendage presence or absence in stem members of the family. Without direct fossil evidence of cranial appendages in early musk deer relatives, the question of loss versus absence remained open. However, the overall pattern across ruminants showed a clear trend toward cranial appendage elaboration in most lineages, making secondary loss in Moschidae plausible even without direct fossil confirmation. The discovery that even closely related families could differ so dramatically in cranial appendage presence underscored the evolutionary lability of these structures. They could be gained, elaborated, or completely lost, depending on selective pressures. Recent discoveries of fossil ruminants showing rudimentary versions of cranial appendages provided additional support for gradual evolution from simple precursors. Stem members of families known for elaborate headgear often possessed simpler, less developed versions, suggesting the complex forms seen today evolved gradually from more basic structures. If each cranial appendage type arose independently, we might expect the earliest members of each family to already possess the distinctive features of their group. Instead, the fossil record showed gradual elaboration, exactly as the homologous core hypothesis predicted. The genetic evidence helped interpret these rudimentary fossil forms. Rather than attempting to classify simple bony outgrowths as proto-horns or proto-antlers based on subtle morphological features, researchers could now understand them as early stages of the shared developmental program that neural crest cells employed to build cranial appendages. The later divergence into covered versus bare, permanent versus deciduous, and continuously growing versus regenerating forms occurred through modifications to this ancestral program. A stem bovid with a simple bony projection and a stem cervid with a similarly simple structure might both represent the primitive condition before their lineages specialized. The extreme diversification of cranial appendage forms within Bovidae alone demonstrated the evolutionary plasticity of these structures. Horns range from the simple, slightly curved protrusions of gazelles to the massive, spiraling horns of greater kudu to the broad, curved horns of water buffalo. All bovids build their horns through the same basic developmental process, yet mutations affecting growth rates, directions of curvature, and final dimensions generate spectacular morphological variety. If such diversity could evolve within a single family sharing a recent common ancestor, the differences between horns, antlers, and ossicones across families with older divergence times become less surprising. Sexual dimorphism in cranial appendages added another layer of complexity. In many species, males develop larger, more elaborate structures than females, and in some cases, females lack cranial appendages entirely. This sex-specific development implied that the genes controlling cranial appendage growth respond to hormonal signals, particularly testosterone and related androgens. The hormonal sensitivity made sense given the role of these structures in male-male competition for mates, but it also raised questions about the ancestral condition. Did the common ancestor of all ruminants possess cranial appendages in both sexes or only in males? The widespread presence of cranial appendages in females across multiple ruminant families suggested the ancestral state included both sexes bearing these structures, with male-only development evolving later in certain lineages. However, interpreting sexual dimorphism patterns required careful phylogenetic analysis, and the sparse fossil record for many groups left considerable uncertainty. The developmental timing differences between horns and antlers also demanded explanation. Horn buds in cattle differentiate during the second month of fetal development, though actual bone growth begins only after birth. In contrast, most cervids do not begin pedicle or antler development until puberty, linking cranial appendage growth to sexual maturity. This dramatic difference in timing challenged simple homology hypotheses that assumed shared structures should follow similar developmental schedules. Yet timing differences need not indicate separate origins. Regulatory mutations that alter when genes are expressed can shift developmental events earlier or later without changing the fundamental nature of the structure being built. The phenomenon of heterochrony, evolutionary changes in developmental timing, has generated much of the morphological diversity seen across animals. A mutation that delayed cranial appendage development until puberty could have occurred in the cervid lineage without fundamentally altering the underlying genetic program inherited from the common ruminant ancestor.
The broader implications extended beyond ruminants to our understanding of how development and evolution interact to generate morphological diversity. The cranial neural crest represents one of the most important innovations in vertebrate evolution, enabling the diversification of head shapes and feeding strategies that characterizes this group. The co-option of neural crest cells to build cranial appendages demonstrated how existing developmental programs can be modified to create novel structures without inventing entirely new genetic machinery. The same cells that construct jawbones and facial structures could be recruited to grow defensive and display ornaments on the skull. What remains unknown, however, is which specific genetic changes differentiate horn development from antler development. Somewhere in the genomes of bovids and cervids lie mutations that determine whether a cranial appendage will be permanent or deciduous, covered in keratin or velvet, growing continuously or cycling annually. The gene expression studies identified patterns supporting horn-antler homology, but did not pinpoint the precise genetic differences. Finding these mutations would transform our understanding from recognizing that horns and antlers share common origins to explaining exactly how their developmental programs diverged. What evolutionary tinkering with regulatory networks produced a structure that sheds annually instead of growing continuously, or that wraps itself in keratinized skin instead of velvet? The evolution of hoofed mammals reveals a story far richer than anyone imagined. These animals walked with dinosaurs 20 million years earlier than we thought, survived extinction through climate-driven pulses of diversification, transformed so completely that some abandoned land entirely to become whales, and built their spectacular array of horns, antlers, and ossicones from a single shared developmental program hidden in their genes. What emerges is a fundamental truth about evolution. It works not by creating entirely new structures from scratch, but by endlessly modifying what already exists. The same neural crest cells that build a deer's face also construct its antlers. The same hoofed ancestor that gave rise to cattle also produced whales. The asteroid that devastated Earth also set the stage for mammalian dominance through volcanic warming pulses. The next time you see a deer in the forest or watch whales breach offshore, remember, you're witnessing millions of years of evolutionary tinkering, constraint, and opportunity playing out in living form. Thank you for listening.